and welcome to my presentation. My name is Ria, and I will be talking to you today about how host lipids regulate the multicellular behavior of Capsaspora owczarzaki. So you are all aware that animal microbe symbiosis is a widespread phenomenon from plants to insects to sea creatures, and even in people. The microbiome of an animal can have a myriad of effects on its host. These are often arranged in a spectrum from pathogenic to mutualists, and microbes can be acquired in many ways, from horizontal transfer to vertical transfer. In 1979, a mysterious protozoan was isolated from the freshwater snail called Biomphalaria glabrata, and this protist was isolated from multiple types of snails, and it was shown to swarm and kill snail parasites. Now, this protist was later named Capsaspora owczarzaki, and it's thought to be a predictive symbiont of snails, but not much research has been done to understand this relationship since then. Now in 2024, the system is primed for study. Since 1979, many genetic and molecular tools have been developed for Capsaspora, and Biomphalaria has been well studied in the context of schistosomes, which can also infect humans. So our lab decided to further characterize this symbiotic relationship. We looked at what happens inside of snails. We injected Capsaspora into snails, and here we've injected into the mantle and the pericardium, which is where Capsaspora was first isolated. But very quickly, Capsaspora forms multicellular aggregates inside of the mantle cavity and the pericardium. And these aggregates are only present inside of the snail tissue, and the aggregation is calcium dependent. We next extracted hemolymph serum from the snails and tested them for aggregation, and we saw that Capsaspora forms aggregates in response to the serum. But what in the serum causes this activity? We performed several fractionation experiments, which I won't go through here for the sake of time, but this led us to believe that lipids were responsible. We extracted the lipids from snails, and we showed that indeed these induce aggregation. Here's a nice dose-dependent activation, and we've done lots of characterization of these lipid particles, including what kinds of lipids are involved. If we take this lipid information and we make a simple lipid mix out of commercially available sources, we see similar aggregation activity. Of the lipids in the simple mix, called di-oleoyl phosphatidylcholine, was active on its own. And we've now shown that cells will aggregate inside of snails and in the presence of snail serum, and that this activity is due in part to this lipid DOPC. So the next question we asked was, can Capsaspora sense changes in its snail host? It suggests that things like diet and infection with parasites will change the lipids present in their serum. So we infected a group of snails with parasites and extracted the serum from them and tested them for aggregation. Here we've seen that there's dramatically more aggregation in response to snail serum that was collected from infected snails. We quantified the lipids in these samples, and we were able to show that many phosphatidylcholines, including DOPC, are upregulated in the infection state. And so, although this protist was first isolated a long time ago, this has never been reported before. And so if you take nothing else away from this presentation, it's that lipids present in the hosts of snails can regulate the aggregation behavior of Capsaspora. In the future, we are investigating if this behavior is essential to colonize and protect snails from schistosome infection. If you have questions, feel free to email me.